ALDABA. In this video lesson, we will be discussing about glycolysis and the Krebs cycle. But before that, let us all pray.
Lord, as we begin today, we ask freshness of your Spirit, that out of confused issues may come simplicity of plan, that out of fear may come confidence, that out of hurry may come deliberation, that out of frustration may come guidance. Let us get to our work, not head first, but heart first. May we be able to disagree without being disagreeable, to differ without being difficult, to be honest without tension, and frank without offense, in an atmosphere of team spirit. Amen.
For new, we are hoping that the students will learn to understand the topic about glycolysis and the Krebs cycle. Before we start our topic, I have here some activity. What is glycolysis? Glycolysis is an anaerobic reaction which occurs in the cytoplasm of the cell. Good. What is the importance of glycolysis? Glycolysis is important in the cell because glucose is the main source of fuel for tissues in the body.
Good day everyone. Today I will going to discuss to you about the glycolysis, pyruvate oxidation, and the Krebs cycles. When we say glycolysis, it is the process of splitting a 6-carbon glucose molecule into 3-carbon pyruvate molecules is linked to the Krebs cycles. So it means the Krebs cycles and glycolysis have a same process of splitting with the same carbon with 6 carbon. It is also the glucose molecule respond to the cycle reactions occur twice. So it means not only for one but also thrice the cycles. It is also 2 pyruvate acid molecules are formed. In the Krebs cycles, there are functions and products about it. For the one cycles, 2 molecules of an carbon, 3 molecules of NAD or NADH, 1 molecule of AD2, and 1 of the molecule of ATP or adenosine triphosphate, molecules of acetyl-CoA enough to the 2 cycles. It is very important to us to know what is the importance of Krebs cycles and the glycolysis, because it can apply as what is the respirations of this matter, and these products will gain us for any oxidations that respired. Though they only want ATP, but it is only can produce ATP or adenosine triphosphates. And when process of respiration, that is called oxidation phosphorylations. So it means pag nag-respiration niya, the only one thing is ATP, the NADPH or NDP or ATP or adenosine triphosphate. That's all thing.
Krebs cycle comes from lipids or fats and carbohydrates, which both produce the molecule acetyl coenzyme A. The citric acid cycle, also known as the Krebs cycle or TCA, is a series of chemical reactions to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins. The Krebs cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats, and proteins into carbon dioxide. The Krebs cycle involves a series of enzyme-catalyzed reactions that reduce the acetyl portion of acetyl coenzyme A in the mitochondrial matrix. In the first step of the cycle, acetyl CoA combines with 4 carbon acceptor molecules, oxaloacetate, to form a 6-carbon molecule called citrate. After a quick rearrangement, these 6-carbon molecules releases 2 of its carbons as carbon dioxide molecules in a pair of similar reactions, producing a molecules of NADH enzyme. The enzymes that catalyze these reactions are key regulators of the citric acid cycle, speeding it up to slowing it down based on the cell's energy needs.
Here are the steps in the Krebs cycle. In the first steps of the citric acid cycle, acetyl CoA joins with a 4-carbon molecule, oxaloacetate, releasing the CoA group and forming a 6-carbon molecule called citrate. In the second step of the citric acid cycle, citrate is converted into its isomer or isocitrate. This is actually a 2-step process involving first the removal and then the addition of a water molecule, which is why the citric cycle is sometimes described as having 9 steps rather than the 8 listed here. In the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule, a ketoglutarate. During these steps, NAD reduced to form NADH. The enzyme catalyzing these steps, isocitrate dehydrogenase, is important in regulating the speed of citric acid cycle. The fourth step is similar to the third. In this case, it is a ketoglutarate that oxidates, reducing and releasing a molecule of carbon dioxide in the process. The remaining 4 carbon molecules pick up coenzyme A, forming the unstable compound of succinyl. The enzyme catalyzing these steps, a ketoglutarate dehydrogenase, is also important in regulation of the citric acid cycle. In step 5, the CoA of succinyl is replaced by a phosphate group, which is then transferred to make ATP, and in some cells, GDP guanosine diphosphate is used instead of ADP, forming GTP guanosine triphosphate as a product. The 4-carbon molecule produced in this step is called succinate. In step 6, succinate is oxidized, forming another 4-carbon molecule called fumarate. In this reaction, 2 hydrogen atoms with their electrons are transferred to FAD, produced FADH. The enzymes that carries out the step is embedded in the inner membrane of the mitochondrion, so FADH can transfer its electrons directly into the electrons transport chain. In step 7, water is added to the 4-carbon molecule fumarate, converting it into another 4-carbon molecule called malate. In the last step of the citric acid cycle, oxaloacetate, the starting 4-carbon compound, is regenerated by oxidation of malate. Another molecule of NAD is reduced to NADH in the process.
For more information, we prepared a short video for the clarification of the topic.
The Krebs cycle is the second step of cellular respiration. The Krebs cycle needs oxygen in order to proceed, and it takes place in the inner matrix of the mitochondria, which are found inside cells. If oxygen is present, pyruvate that is formed in the first step of cellular respiration is taken into the inner membrane and the Krebs cycle begins. During the preparatory stage of the Krebs cycle, pyruvate molecules left over from glycolysis give off a carbon atom in the form of CO2. Next, the Krebs cycle proceeds. The newly formed 2-carbon molecule combines with a molecule called coenzyme A. This produces a new product that is important to the Krebs cycle called acetyl-CoA. The reactions required for producing acetyl-CoA store an electron in a molecule called NADH that is used later during cellular respiration. The Krebs cycle begins when a 4-carbon molecule is combined with acetyl-CoA, forming a 6-carbon molecule called citric acid. Next, during a chain of reactions, 2 carbons are given off from citric acid in the form of CO2. 1 ATP molecule is produced. And since high-energy electrons are produced during this chain of reactions, 3 NADH molecules are produced. When NAD+ takes on an electron, it becomes NADH. So, 3 NADH molecules are produced. When 2 electrons are stored in a molecule called FAD during the Krebs cycle, it becomes FADH2. This chain of reactions produces 1 FADH2 molecule. We care about these stored electrons because they are used during the electron transport chain portion of cellular respiration, which occurs directly after the Krebs cycle. The Krebs cycle produces a lot of important molecules. When we started the Krebs cycle, we followed the story of one of the pyruvate molecules that was produced during glycolysis. But glycolysis actually produces 2 pyruvate molecules, so we'll have to double the amount of products produced by the Krebs cycle in order to have an accurate estimate of how much energy cellular respiration can produce from just 1 glucose molecule. The grand total for the Krebs cycle comes to 2 ATP, 6 NADH molecules, and 2 FADH2 molecules. The electron storing that has taken place during glycolysis and the Krebs cycle are used in the third and final stage of cellular respiration, the electron transport chain.